Hi students, welcome to Nucleus Neet Coaching. So every day in the class, it will be boring. No teacher will make any fun, right? But today, let's have some fun in our class. So, I'm going to play a song, try to identify the song, okay? One of the most famous melodies in Bollywood. How was that? Amazing, right? How many of you are fans of Shah Rukh Khan? Are you a fan of Shah Rukh Khan? I am the great fan of Shah Rukh Khan. Na 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 na. Amazing. क्या गाना है यार? Amazing ना? So now let us start the class. So today's topic is ribosomes. What are ribosomes? Ribosomes are granular structures which were first observed by a scientist called George Plastids inside the cells. If you observe a cell, in the cell, under the electron microscope, you can see granular structures. These granular structures, they were identified as ribosomes by a scientist called George Plastids. So these ribosomes, actually they function as protein factories. That's why they are called protein factories of cell. Generally in a factory, what will happen? Some manufacturing process will be taking place. In this ribosome, which is called protein factory, protein manufacturing will be taking place. Proteins will be manufactured. Proteins will be synthesized. That's why the name protein factories of cell. Okay. Next. The ribosome is composed of which molecules? It is composed of ribonucleic acid and proteins. Ribosomes are made up of ribonucleic acids, RNA and proteins. Both of them will be present inside the ribosomes, and ribosomes are non-membrane bound cell organelles. That means there is no membrane covering the ribosome, just like as in the case of chloroplast, mitochondria and vacuole. There some membrane was there, but here there is no membrane. That's why they are non-membrane bound organelle. Can you tell one more non-membrane bound cell organelle? Nucleolus. Nucleolus also does not have any outer membrane. That's why nucleolus and ribosome both are non-membrane bound cell organelles. Note down this point. Next. The ribosomes, they synthesize proteins by a process called as translation. So what is the process called? Translation. Translation is a process which is undertaken by ribosomes to synthesize proteins. Okay. So here ribosomes, when they are in unassembled state, there will be two subunits. One is the large subunit and another is the small subunit. They will be separately present in the cytoplasm. So when the ribosomes need to assemble at that time, the large subunit and small subunit, they come closer and attach tightly like this. Okay, that results in formation of what is called your ribosome. So when the small and large subunit will bind together, there will be a small opening. Okay, there will be a small opening or a cleft. In this opening, what happens? The protein synthesis process will take place. And here I can show you. So this is the large subunit, this is the small subunit. Large subunit and small subunit, when they assemble, they form a ribosome, which is called the protein factory of the cell. So in the small cleft which is present between these two units, protein synthesis process will take place. As a result, a chain of amino acids will be formed. A chain of amino acids will be formed. So this chain of amino acids which is formed is called polypeptide chain. Come on students, tell me what it is called. Polypeptide chain. Polypeptide chain. Each round which I have represented here, each round represents one amino acid. So several amino acids, when they are attached end to end, then it forms a long chain. It is called polypeptide chain. This polypeptide chain, it will undergo certain changes inside the endoplasmic reticulum and Golgi complex, and further it gets converted into a functional mature protein. Okay, so that is the function of ribosome. Okay, now we will discuss about the different types of ribosomes. In the living cells, we can see two types of ribosomes. 70S ribosomes. 70S ribosomes and 80S ribosomes. Two types of ribosomes only we can see in the living organisms. 70S ribosome is made up of two subunits. The large subunit is called 50S subunit, whereas the small subunit is called 30S subunit. Both of them, when they combine, they give rise to 70S ribosome. Okay, 70S ribosomes are found in all the prokaryotes, present in the chloroplasts, cell organelles and mitochondria in the eukaryotes. Okay, whereas the 80S ribosome, it is having the large subunit called 60S, small subunit called as 40S. Both of them, when they combine, they give rise to 80S ribosome. Okay, so 80S ribosomes are present in all the eukaryotes, in all the eukaryotic cells. Okay, then what is the meaning of S here? What does S stand for? S stands for Swedberg unit. The scientist who invented this unit is called Swedberg. That's why S is called Swedberg unit. And what does it represent? It represents the sedimentation coefficient of ribosome. What is sedimentation coefficient? It is a measure of density and size of ribosome. Understood, students? So in the prokaryotes, we have seen that several 70S ribosomes, when they are attached to messenger RNA, they form a structure called polysome. They form a structure called polysome. Polysome is also called polyribosome. This point we have already discussed. Okay, the function of polysome or polyribosome is nothing but protein synthesis. Whereas in eukaryotic cells, imagine this is a eukaryotic cell, this is a nucleus, this is the rough endoplasmic reticulum. On the rough endoplasmic reticulum, you can see these granular structures called ribosomes. So these ribosomes which you can see on rough endoplasmic reticulum are 80S type. 80S type. They are also involved in protein synthesis. Students, now we will discuss another very important cell organelle in eukaryotic cell, that is mitochondria. Mitochondria is also called powerhouse of the cell. Can you imagine why a mitochondria is called powerhouse of the cell? I will tell you the reason. Imagine that in our apartment or in our colony, during night, suddenly there is a power cut. Then what will happen? Everywhere it will become dark. We will not be able to see. So someone will go and switch on the generator in the powerhouse, and when generator is switched on like that, again we will get power. Okay, so just like a powerhouse which provides electricity to all the houses in the apartment, so inside the cell also there is a powerhouse who is none other than mitochondria that will provide energy to the entire cell, and that's why mitochondria is called powerhouse of the cell. Understood? Understood? Now let us discuss about the shape of mitochondria. So inside the cell, the mitochondria, it provides energy to the entire cell. A mitochondria has sausage shape. So what is the shape of mitochondria? It has sausage shape. So what is a sausage? A sausage is a bakery item, a food item which you can see here. This is the sausage. It is having sausage shape and it is having a diameter ranging from 0.2 to 1 micrometer. 0.2 to 1 micrometer is the diameter of each mitochondria. Okay, now what is the function? As we discussed, it provides energy to the cell. But how a mitochondria will provide energy to the cell? Simple. It will take part in aerobic respiration. What is aerobic respiration? It is a method, it is a process, metabolic process, by which the mitochondria can produce ATP energy by consuming oxygen. That's why it is called as aerobic respiration. So it is a process, metabolic process, by which the mitochondria can release ATP energy by consuming what? By consuming oxygen. So that's why it is called as aerobic respiration. Okay, next, coming to the actual shape of mitochondria. The mitochondria has a double membrane bound structure. It is covered by two membranes. Okay, so now let us see the structure of mitochondria. This is the sausage shape. Okay, what you are seeing here is the outer membrane, which is continuous. This is the outer membrane of mitochondria. Below the mitochondria, you can see the inner membrane. The inner membrane shows number of folds. What are these folds called? These folds are called cristae. These folds are called as cristae. You can see here, these are the folds. In which membrane you can see the folds? You can see in the inner membrane. Okay. This is the sausage shape. So it is just like a mitochondria which is sliced and opened. Okay. This is the image of mitochondria, diagram of mitochondria, which gives the inner structure. Okay. If you cut open the mitochondria, it looks like this inside. So this is the outer membrane. This is the outer membrane of mitochondria. It has several enzymes which are required for the functioning of mitochondria, and this is the inner membrane. Inner membrane. Okay, the outer membrane is continuous, whereas the inner membrane has several infoldings. So what are these infoldings called? These infoldings are called cristae. What they are called? They are called cristae. Understood, my children? Now look here. Inside the mitochondria, you can see there is a fluid. This is fully filled with a fluid. So what is this fluid called? This fluid matrix is called matrix. It is called as matrix. Okay, the fluid present inside the mitochondria is called as matrix. Now in the matrix we can see several enzymes. Many enzymes are present which are involved in aerobic respiration, and some enzymes are involved in protein synthesis. Okay, so there are different varieties of enzymes in matrix that are involved in aerobic respiration and protein synthesis. Okay, note down this point. Now these cristae have tennis racket shaped particles. All over. I am just drawing on one cristae. So you have to imagine all the cristae will have also this tennis racket shaped particles. These tennis racket shaped particles are called as oxysomes. Okay, they are also called F0, F1 particles. F0, F1 particles. They play very important role for synthesis of ATP, that is adenosine triphosphate. What is the full form of ATP? Adenosine triphosphate. It is also called the energy currency of the cell. Energy currency of the cell. So that is about oxysomes. Okay. So look here students, it is a double membrane bound cell organelle. The mitochondria, it divides by process called as fission. It divides by process called as fission. That means one mitochondria, it divides into two mitochondria. We call that process as fission. Next, the number of mitochondria depends upon physiological activity of the cell. Here, inside the cell, there may be 2 mitochondria, or there may be 4, or there may be many mitochondria. So the number of mitochondria in a eukaryotic cell, it depends on what? It depends upon the physiological activity. Okay, how much active the cell is, based on that the number of mitochondria depends. If cell is very active in producing different types of materials, then obviously there will be more mitochondria. If the cell requires more energy, then obviously there will be more mitochondria inside the cell. Okay, so that is the relation. The outer membrane is continuous as we discussed, and the inner membrane has several infoldings which are called as cristae, and oxysomes are present in the cristae. These are oxysomes. Understood, my dear students? So this is about mitochondria and what type of enzymes are present in the matrix. In the matrix there are enzymes. Matrix has enzymes required for aerobic respiration and protein synthesis. This is another important point for your NEET examination. Now what else is present in the matrix? Let's see. In the matrix you can also see circular DNA. There is a circular DNA, and also you can see RNA. You can also see RNA, ribonucleic acid, and you can also see ribosomes. Cell organelles called as ribosomes. What type of ribosomes you can see? 70S type of ribosomes present inside the mitochondrial matrix, which are involved in protein synthesis. So this is about mitochondria, students. Copy down. Now we will discuss another type of cell organelle called as plastids. Plastids are just like mitochondria, they are also double membrane bound cell organelles, and they are found in plant cells and also in euglenoids. They are found in plant cells and also euglenoids. What are euglenoids? In kingdom Protista we have discussed this type of organisms. This type of unicellular organisms are called as euglenoids. In these euglenoids also you can find plastids. Okay, so they are present in plant cells and also euglenoids. Note down this point. So these plastids, they have varieties of functions in plant cells. Based upon the photosynthetic pigments, these plastids are classified into 3 types. They are chloroplasts, chromoplasts and leucoplasts. So based upon what? Based upon the pigments that are present in these organelles, they are divided into 3 types. Now let us see the classification of plastids. Plastids, based upon the type of pigments, they are classified into 3 types. Chloroplasts. Chloroplasts are green plastids. They are green in color. They are green plastids. Their main function is they are involved in photosynthesis. Okay. Next, the second type of plastids are chromoplasts. chromoplasts. Chromoplasts are colored plastids. Colored plastids. They have varieties of colors. They exist in different colors. They may occur in red, they may occur in orange, or they may occur in yellow color. Okay, so these chromoplasts, what is their function? They impart bright colors to petals of the flowers. That is the function of chromoplasts. They are not involved in any photosynthetic activity. The third type of plastids are leucoplasts. Leucoplasts. Leucoplasts, they doesn't have any pigment. That's why they are colorless. They are colorless plastids. So all these plastids are double membrane. They are having two membranes. This is the outer membrane and inner membrane. Both membranes are continuous. They doesn't show any foldings. But based upon the pigments, based upon what? Based upon the pigments only, they are of three types: chloroplasts, chromoplasts, and leucoplasts. Okay. So now what pigments are present in chloroplasts? We will discuss. The pigments which are found in chloroplasts are as follows: chlorophyll, which is obviously a green color pigment, which imparts green color to the leaves, chlorophyll pigment. In addition to this chlorophyll, there is also carotene pigment, which is orange color pigment, and also xanthophyll pigment, which imparts yellow color to the Okay, so these are the three pigments which are present in the chloroplast. You may ask one doubt: even though there is carotene and xanthophyll which are orange and yellow color, why the leaves appear in green color, chlorophyll? The reason is that chlorophyll is the dominant pigment. It is the pigment which is present in large amount in the leaves. That's why leaves appear in green color. So now what are the pigments that are present in chromoplasts? Pigments that are present in chromoplasts. Chromoplasts, they doesn't have chlorophyll pigment. There is no chlorophyll pigment, but there is carotene, which can impart orange color, and there is xanthophyll, which can impart yellow color to the flowers. Okay, in addition to this carotene and xanthophyll, other pigments are also present, other soluble pigments are also present, which provide variety of other colors like blue color, purple color, etc., etc. Okay, other pigments are also there, but in the context of NCERT, we no need to learn about the other pigments, just other pigments. Some more pigments are also present in the chromoplasts. Okay, next, leucoplasts, as the name itself tells that they are colorless plastids. Their function is to store the food material. Based upon the type of food material they store, these leucoplasts are further divided into three types. What are the three types of leucoplasts? Now let us learn. The first type of leucoplast, which stores starch, they are called amyloplasts. Amyloplasts, they store starch as food material. Amyloplasts, they are present abundantly in the potato. So if you cut the potato, in the potato, the white material which you can see, it is full of amyloplasts. Okay, next, second type of leucoplasts, elaioplasts are plastids that store oil. You take a groundnut or coconut, okay, inside the groundnut seed cotyledon you can see lot of oil, groundnut oil. So the cells present in the groundnut cotyledons contain elaioplasts that store oil. Okay, all the oil seeds are rich in elaioplasts. Okay, next is Aleuroplasts. Aleuroplasts. Aleuroplasts are otherwise called proteinoplasts because they store protein as a food material. They store protein as a food material. Aleuroplasts are present in a layer called aleuron layer, which is present in the monocotyledonous seeds. Okay, so these are the different types of plastids. Plastids are three types based upon the pigments. Green colored plastids are called chloroplasts. They are found in green parts of the plant such as leaves. Chromoplasts, they are the plastids which provide different bright colors to the flowers of the plant such as red, orange, yellow. Next is leucoplasts. They are colorless, they doesn't have any pigments but they store food material. So what is their function? They store food material. So based upon the type of food they store, they are again divided into three types. A myeloplast store starch, example is potato. Example is potato. They are present in potato. Elaioplasts are the plastids which store oil. Example is your oil seeds like groundnut, sunflower. Aleuroplasts, they store protein. Example, the aleuron layer. Aleuron layer of monocot seeds. Okay, so this is about plastids. Now we will discuss in detail about chloroplast. Note down this, students. Students, next cell organelle is chloroplast. Chloroplasts are called as kitchens of the cell. Just like kitchen in our house our mother will prepare nice yummy food for you, right? So just like a kitchen in our house, inside the cell also there is a kitchen which is called chloroplast. The chloroplast will prepare the food that is required for the plant cell. Okay, so now let us discuss in detail about chloroplasts. The chloroplasts are mainly found in mesophyll cells of leaves. They are found in which cells? Mesophyll cells. Inside the leaf there is a kind of tissue called mesophyll tissue. The cells which are present in the mesophyll tissue are called mesophyll cells. So in those cells called mesophyll cells, we can see these green colored plastids called as chloroplasts. These are the chloroplasts. They will be arranged like this in the mesophyll cells of leaves. Next.
Students, here you can see the tiny chloroplasts are moving slowly along the inner walls of the mesophyll cells along with the cytoplasm. This movement is called cyclosis, otherwise called cytoplasmic streaming movement. It is brought by cytoskeleton proteins. So this movement will allow the chloroplast to distribute the nutrients uniformly throughout the cytoplasm in the cell and also help them to escape high intensity of sunlight. So this is about cytoplasmic streaming movement.
What is the length of each chloroplast? The length ranges from 5 to 10 nanometers and the width is ranges between 2 to 4 nanometers. Just like mitochondria, they are also double membrane bound cell organelles. They are covered by two-layered membrane. Now let us see the diagram of a typical chloroplast. Generally in the plants you can see lens-shaped chloroplast. Lens-shaped chloroplast I am going to draw now. Look here. So this is a lens-shaped chloroplast which we can see in the mesophyll cells of plants. In the mesophyll cells of plants. There are two membranes, outer membrane and inner membrane. So this is the outer membrane. This is the outer membrane. This is the inner membrane. Let me make some adjustments. Yes, this is the outer membrane. Outer membrane. The inner membrane. Below the outer membrane there is inner membrane. So these are the two membranes of chloroplast. If you compare mitochondria and chloroplast, you can see one difference. Our mitochondria, it was in sausage shape. Here the chloroplast in plants, it is in lens shape. In mitochondria, the inner membrane, it had several infoldings, but here the inner membrane does not show any infoldings. Got? So let me make my diagram little bit perfect. Yes. Is it perfect? Okay. Now, there is a liquid matrix inside the chloroplast. Now, this liquid matrix is called stroma. Stroma. What is the liquid matrix present in the chloroplast called? It is called as stroma. Okay. In the stroma, you can see several sac-shaped structures. You can see several sac-shaped structures called as thylakoids. So you can see here, this is a sac-shaped structure which is called thylakoid. So several thylakoids are arranged one over another. They are piled one over another like this. When they are piled one over another, each unit is called as granum. Each unit is called as granum. So each individual structure is a thylakoid. Several thylakoids, when they are arranged one over another like piles of coins, then this entire structure is called granum. Granum. Grana is plural, granum is singular. Okay. So in the thylakoid membrane, you can see different types of photosynthetic pigments that take part in photosynthesis. So in this thylakoid membrane, you can see chlorophyll pigments, xanthophyll, carotene. So these photosynthetic pigments are present in this thylakoid membrane. Understood? I am showing in blue color. So these are the pigments, they are present in the thylakoid membrane. Okay. So the space which is present in each thylakoid, this space is called as lumen. What it is called? Lumen. The space which is present in each thylakoid. The space present inside the thylakoid is called lumen. L-U-M-E-N, lumen. Okay. So like this, many grana are present in the stroma. So let's draw here one more granum. Thylakoid, another thylakoid, another thylakoid, and one more, another thylakoid. Here, let us draw one more granum. This is a thylakoid, another thylakoid, one more, and one more. Last one. So like this, several grana will be distributed inside the stroma of the chloroplast. Now, adjacent grana will be interconnected by tubular structures which are called as stroma lamella. So this is a stroma lamella. Now these two grana are interconnected. This is also stroma lamella. This tube-like membranous structure is called stroma lamella. They will interconnect the adjacent grana with each other. Okay. So the main process of photosynthesis, it is taking place in the grana, and also a part of the photosynthesis is also taking place in the stroma. So other than this grana, what else you can find in the stroma? You can find circular DNA. You can find circular DNA just like mitochondria, and there is also RNA, ribonucleic acid. You can also find ribosomes. What type of ribosomes? 70S ribosomes. 70S ribosomes are also present in the chloroplast. So these are all things which you can find in the chloroplast. Okay. Now let us briefly discuss how a chloroplast will work. So when sunlight falls on the chloroplast, what happens? The photosynthetic pigments, chlorophyll pigments, they will capture the light energy and convert the light energy into chemical energy, that is ATP and NAD. Okay. So these chemical energies later they will be used in the stroma to convert the atmospheric carbon dioxide into the food material, that is glucose. Okay. So the glucose will be produced in the stroma by using carbon dioxide. Okay. So the glucose which is formed inside the in the stroma, it will be later converted into the storage form of food material called as starch. In the form of starch. So this storage form of food material called starch, it will be stored inside the chloroplast in the form of starch granules. Starch granules. So in this way, the chloroplast can prepare the food material. Okay students, understood everyone? So look here, some more points we'll discuss. The shape of the chloroplast. Some chloroplasts occur in lens shape, some occur in oval shape, some are spherical, some are discoid, some are ribbon-like. These are different possible shapes of chloroplasts. The inner membrane is less permeable. The inner membrane is less permeable. It will allow less number of molecules to enter into the chloroplast, whereas the outer membrane, it is more permeable to molecules. Okay. Next, the thylakoids are membranous sacs. They have pigments. So these are thylakoids. They are membranous sacs which possess photosynthetic pigments like chlorophyll, carotene, xanthophyll, etc. And thylakoids form piles called grana. Several thylakoids, when they are stacked one over another, they form structures called as grana. Okay. Next, stroma has enzymes required for carbohydrate and protein synthesis. In the stroma, you can find enzymes that take part in two processes. One is carbohydrate synthesis and another is protein synthesis also. Okay. So these are some of the points. Copy down students. One more important point, in the mesophyll cell. In the mesophyll cell, the number of chloroplasts will range from 20 to 40. 20 to 40 per each cell. Another important point. Okay.
Students, my name is Kiran Kumar. I have more than 10 years of experience in teaching NEET biology to the students, and I know the techniques to make an average student into a topper student. If you are looking for getting more than 600 marks in the NEET examination, then subscribe our channel. We'll continue the class in the next video. Until then, take care. Bye.